Hi everybody, DrBiology here, and this is a video on gene mutation, and it's related to, you can actually see it, 3.4.3, genetic diversity can arise as a result of mutation or during meiosis. So let's just have a quick recap of protein synthesis. So first of all, we have a DNA, and the DNA is a template, and there's a template strand there, okay? And transcription occurs where mRNA is produced, pre-mRNA I should say, and then it is spliced to ensure that the introns and exons are separated, and then it goes off to the ribosomes and translation occurs, and that's where you get your string, your sequence of amino acids joined together by peptide bonds to make a polypeptide. Previous video of mine, I've got a really good animation of protein synthesis, so please do check that out. Right, so what is gene mutation? Well, it does involve changes in the DNA base sequence of chromosomes. So types of errors that can include, first of all, substitution. So when one base is substituted with another. So here we have a genetic code, and you can see the codons, the triplet code, and what it actually codes for. So these are the different amino acids down here. So let's see what happens when you substitute one base with another. So let's see what happens when this third adenine base it changes. So watch what happens. So you can see it changes, and it means that the amino acid is changed, which is leucine. So you're probably thinking, well, what effect does that have on the final polypeptide? Well, sometimes it has no effect. So it's what we call a silent mutation, because if it's on the third base of a codon, it may have no effect because the third base doesn't affect the amino acid coded for all of the time. So all codons beginning with CC code for proline, for example. Or it could be a nonsense mutation where the protein is non-functional. So you might get a premature stop codon, means the protein isn't finished, or it's a non-functional protein because of the sequence of amino acids. So let's have a look at deletion. So deletion changes in an individual gene due to the miscopying of one or more nucleotides. So it can be either deletion of a base or addition of a base, and it results in what we call a frame shift. So let's have a look to see what happens with the frame shift. So you can see here, so CGA, let's get rid of the A, and you can see then it moves across and it changes these two first amino acids, and that can have pretty serious consequences. So they change the codon reading frame, even though they don't change the actual sequence of the bases sometimes. So all amino acids downstream from there are wrong, and the protein is therefore non-functional. So remember, the code is degenerate. So the degenerate nature of the genetic code means that some amino acids are coded for by more than one DNA triplet. So you can see a substitution here of from T to C, but that has no effect whatsoever on the final sequence because it's on the third part of the codon. However, deletions do change the number of bases present, and that will cause a frame shift and all the bases triplets after it. So you can see A is missing, so therefore it will shift, and therefore you'll get GTC, so it will code for valine rather than serine. Hope you found that useful. Please do subscribe to DrBiology, and I'll see you soon.